Tonight's lecture is going to be on pathways for vision and hearing. If at any point you guys can't hear me or you have any problems, you can use the chat box. Just let us know what's going on. David is also in here tonight. He's another tutor, so he's going to be helping us out. So just a quick introduction. Like I said, we're doing pathways for vision and hearing tonight. My name's Chris. I'm one of the MCAT tutors here as well. And this is actually week 5 of our 15-week MCAT course program. So I just put some contact information up here. This is the link to our GroupMe. If you don't have it already, you can use it. If you want to send it to a friend that you know, definitely do that. I put our Instagram handle over here as well. And I linked you to the YouTube channel where you can get all of the videos that we upload after the lecture. So we'll be sending them, we'll be sending the PowerPoints in the GroupMe after every lecture. And we also have a shared OneDrive where you can access them, and you can get the videos on YouTube. So we do 3 lectures a week. For those of you who are new, it's Thursday, Friday, and Saturday. It's usually from about 8:30 p.m. Eastern time to about 10:30. And then every Sunday we do unstructured office hours where you can just come with your questions if you have anything, or if you just want to come observe and see anybody else's questions. So with that being said, let's dive in. So this is vision and hearing in the brain. So I put a picture over here of the division of the brain, of the cortex specifically. It's divided into 4 lobes, as I'm sure you guys know. We have the occipital lobe over here in the back, which we're going to be talking about. We have the temporal lobe. There's actually 2 parts of it. There's one on each side, which we'll be talking about also. And then there's the parietal and frontal lobes, which we won't be talking about too much today. But you should definitely know them. So how hearing is processed in the brain. So the auditory nerve is a branch of the vestibulocochlear nerve. The vestibulocochlear nerve actually branches off into two parts of the nerve. One part goes to the cochlea, and the other one goes to the semicircular canal, which we'll be talking about a little bit later. That's more important in your vestibular sense and like your balance and coordination. We're going to talk about that a little bit later. And it's excited when hair cells are stimulated. So this nerve goes all the way up to the brain, and it actually leads directly to the thalamus. The thalamus is sort of like the relay center for any of your incoming sensory and motor information. So it processes all sensory information except smell. That's just a little thing that you probably should know. It does 4 out of the 5 senses. Everything goes through the thalamus. So for those of you just joining, I'm just going to ask you to keep your mics muted for most of the lecture. And at the end of each section, I'll give you guys a chance to unmute if you have any questions. And if you have any other questions while I'm speaking, you can put them in the chat box and I'll do my best to address them, or David will address them in the chat. So the thalamus then sends the information to the temporal lobe for higher processing. And like we said before, the temporal lobe is part of the cortex. It's part of your cerebrum. The temporal lobe processes auditory information and olfactory information, auditory being hearing, olfactory being smell. And they house structures like Wernicke's area and Broca's area. So they're also involved in memory and emotion processing, I should say that too. But I know there are some recent studies that have actually kind of said Wernicke's area and Broca's area are a little bit more complex, and they're not as black and white as just be broken down as comprehension and production. But in terms of the MCAT, any sort of study material, anytime I've ever seen it pop up on a test, you should really know that Wernicke's area is primarily involved in speech comprehension, understanding the speech of others, and Broca's area is really mostly involved in the production, you actually physically producing your speech. So those are 2 pretty important areas. I would definitely know them. This lecture, I don't think is super hard conceptually, but there's a lot of vocabulary and there's a lot of terminology. So if you need any clarification regarding that terminology or anything like that, please put it in the chat. Does anybody have any questions about anything on this slide before we move forward? Okay. And how vision is processed in the brain. So visual information is sent to the brain from the eyes via the optic nerve. So it's another one of the nerves that connects your senses to your brain. So the optic nerve brings this information to the thalamus, because like we said before, the thalamus is pretty much the first stop in the brain for this incoming sensory information, everything except smell. So the specific part of the thalamus that the optic nerve connects to is called the lateral geniculate nucleus. Sometimes you'll see it as LGN. I have seen this appear a couple of times in some of my studies. A lot of times it'll appear in a passage, but it's a very good idea to have an understanding of what this is. And then the information from the thalamus travels to the occipital lobe for higher processing, just like the information that comes in from hearing, it first stops off to the thalamus and then it travels to the temporal lobe for higher processing. They're like association areas, and you integrate it into, a higher sort of perception. So the visual cortex houses specialized neurons that detect specific features of incoming information. So a lot of the sensory information that your eyes take in, it's just raw information. It doesn't really mean anything. It's just an image until it gets passed up to your thalamus and then into your occipital lobe for that higher processing. Then there are these specialized neurons that can detect edges, lines, movement. They're all separate and they're all highly specialized for one job. So you have a lot of specialized neurons up there that help you like differentiate stimuli in your environment and just add meaning to the things that you're seeing. So speaking more about the meaning, your cortex processes multiple features and they can integrate memories and prior knowledge simultaneously in a phenomenon called parallel processing. So parallel processing just means that your brain is taking in a bunch of information at the same time, and it is simultaneously processing all of it. And that's what makes up your perception. So for example, if you are looking out your window and you see a bird, you're going to be looking at it and your brain is analyzing the entire picture and it's taking in a bunch of information at the same time. So it's looking at how far this bird is. It's looking at the contrast of the feathers, the color, the intensity of the color, the relative size of the bird. So all of that information is being sent up to your brain. It's being processed at the same time, and it sort of synthesizes one perception or some mental representation. And it also integrates your memories. So that's what top-down processing is. If you have any prior knowledge, if you're an experienced bird watcher, say, and you have some knowledge about the specific type of bird it is, you see red on the bird and oh, that is a red robin. All of that happens at the same time. That's called parallel processing. And that's why humans are so incredible, and that's why we can do what we do with our minds. Does anybody have any questions about that? Parallel processing is a pretty important concept, especially when the MCAT talks about like cogpsych, cognitive psychology. I see that come up a lot in terms of perception. So just make sure you know that. Like I said, we're going to post these lectures. So if you ever want to come back, you can watch the lectures. You can stop, take notes. You can, take notes from the PowerPoints and study that way. Okay. So that brings us to our first review question. So which structure in the brain acts as an early relay center for most of the incoming sensory and motor information? So hopefully you're paying attention. I think I said it a couple of times already. I'll give you guys about a minute and a half to do this. Let me share the poll. Okay, so take a minute to answer this, and then we're going to review it in a minute. And for those of you just joining us, we're just working on our first review question. You could probably access the poll. It's already up if you click the polling button. Please mute your mics, and if you have any questions after each section, you can unmute and talk then, or you can put it in the chat box. All right, I'll give you guys about another 15 seconds, so get your final answers in. Okay. Nice. Okay, that is great. So 92% of you said A, 8% of you said B, and nobody said C or D, or I have no idea. So that's great. The correct answer is, in fact, the thalamus, because like we said before, the thalamus is the first relay center. It's the first stop for incoming sensory and motor information. So it is responsible for relaying 4 out of the 5 senses to the cerebrum, to other parts of your brain for processing. It does not relay your sense of smell. So touch, taste, vision, and hearing all go through the thalamus, and then they get sent to other parts of the brain for further processing. The hypothalamus isn't really involved in any incoming sensory motor information. It's involved in a couple of other things, like homeostatic functions. It's home to the reward center in your brain, but not much with relaying incoming sensory information. So your cerebrum and the different cortices in the cerebrum, they do process sensory information, but they're not an early relay center. That's pretty much like the main event. So that's where the information is trying to go, but the thalamus is really the part of the brain that relays it to the cortex. And the pons is just another part of your brain. It's right by the cerebellum. It's responsible for like balance and coordination, stuff like that. It doesn't really relay sensory information either. Is that clear with everybody? It seems like it was. Anyone have any questions? Very good. Okay, so our next topic is mechanisms of hearing and balance. So we're going to look a little bit more at the structure of the ear and the structure of the eye. Again, like I said before, there's a lot of vocabulary. So throughout the PowerPoint, I just put in bold all of the things that I think you should know, all of the terminology that I think is important. So I would definitely make it a point to either write down the definitions that you don't know. This is even a good lecture for you to use flashcards on, because like I said, most of this is just basic definitions. You're really just, it's memory. Which structure of the brain would serve as the relay center for the smell sense? I am not 100% certain. It's actually funny because I've never seen a question about that on the MCAT. I think it's probably something that a quick Google search would clear up, but I wouldn't worry too much about it. I'm assuming it goes directly to another part of the brain. But in the scope of the MCAT and the scope of this lecture, for now, all you need to know is that 4 out of the 5 go to the thalamus. Okay, so the ear is split into 3 sections. So the external ear, also known as the outer ear, which is right over here to the left, it contains the auricle, which is also known as the pinna, and it directs sound waves into the ear canal. So it's kind of like a satellite dish. It takes the vibrations in the air, the sound waves, and it sort of funnels them in to the ear canal. And the ear canal passes it onto the tympanic membrane. That should not be a C. The tympanic membrane is also known as the eardrum. You're going to find throughout this lecture that almost every single structure here has multiple names. So you should be familiar with both of them. So the auricle is the same as the pinna. Like we said, once it gets to the tympanic membrane, it vibrates, and it directly vibrates on the ossicles. So these little bones in here, excuse me, the malleus, the incus, and the stapes, those are known as the ossicles. So they're really, really tiny bones that are in the inside of your middle ear. The malleus is also known as the hammer. The incus is also known as the anvil. And the stapes, I believe, is known as the stirrup. But I see these 3 more often, so I put those. So like I said, the tympanic membrane vibrates, and it's directly touching the malleus. So you can see right over here on the actual diagram that they're physically touching each other. So these sound waves are now converted into just mechanical vibrations. So this tympanic membrane is actually going to vibrate up on the malleus. The malleus, incus, and stapes are all connected, so the vibration travels through those 3 bones. and then it crosses over to the oval window. So it's directly on the cochlea. The cochlea has a small, it's not listed here, but there's a small part of the cochlea which is called the oval window, and that's actually where the stapes hits the cochlea, and it causes vibration in the fluids inside the cochlea. So some other parts that are not directly involved in the processing of auditory information but are important for the ear that are listed here are the round window, which relieves excess pressure inside the inner ear, and the eustachian tube, also, which equalizes pressure by connecting the ear to the throat. So as I'm sure you know, your ear, nose, and throat are all connected. The eustachian tube is responsible for when your ears pop if you have a change in altitude. That's the eustachian tube equalizing the pressure inside and outside of your body. And you could see like your equilibrium is sometimes thrown off when you get a cold because that tube is sort of crowded. So it's a pretty important structure, and it's definitely noticeable when something's wrong with it. So a little bit more on the cochlea over here. The cochlea is where the mechanical vibrations are turned into electrical impulses. So if we go back over here, you can see this little snail-like structure labeled as the cochlea. It's filled with fluid, and throughout this fluid, there's a membrane that's running through it. It's called the basilar membrane. I have it on another slide as well, so don't worry. And that basilar membrane is covered in these little things called hair cells. So we're going to talk about the hair cells as well. The waves pass through the fluids in the cochlea, and they vibrate the hair cells on the basilar membrane, like I just said. When the cilia on top of the hair cells vibrate, the hair cells release neurotransmitters to the surrounding bipolar neurons, converting sound waves into electric impulses for the cochlear nerve. So if you look right up here, these little cells in pink are the hair cells, and the little projections that are coming on top of them, those are the cilia. So those are super, super fine hair-like structures that protrude into the fluid of the cochlea. So when the ossicles, those three little bones, start banging on the cochlea, it vibrates the fluid, and the fluid moves around, and it starts vibrating these hair cells. So when these hair cells start vibrating, they actually release neurotransmitters. So right at the bottom of these little pink, you see the beginnings of these yellow the yellow neurons, excuse me. So they're actually synapsing with these neurons. So neurotransmitters are released across that gap. And like I said before, these are bipolar neurons. So if you saw my lecture on the nervous system, I said that multipolar neurons are the most common in the nervous system, which is true. But now we're learning about bipolar neurons, and you'll see bipolar neurons a lot. They're always really involved in the reception of sensory information. So we'll see them again when we're talking about vision. But you can see right here, there are two axons. So the information will be passed onto the neuron right over here. It'll travel down this axon. And then this group of bodies is the spiral ganglion cells, because as we know, a ganglion is just a group of cell bodies, soma. And right over here is the auditory nerve. And as we know, auditory nerve is just a bundle of neurons, the bundle of the axons. So that's how, does anybody have any questions about how the mechanical is converted into electrical impulses? You can unmute, you can use the chat box if you like.
Can you start at the outer hair cells? I get everything. Sorry, this is Taja. I get everything from the synapses, like all the way to the ganglions, all that great stuff. But how do you get to the outer cells again? Are those the ones like around the membrane cochlea you said?
Yeah, yeah, absolutely. So the outer hair cells is where it starts. So the outer hair cells, excuse me, are lined throughout the cochlea. That little this little circular structure that we're talking about, it swirls all the way in, and all throughout this there are hair cells, and they're actually ranging in size, which I'll talk about in a minute. But the vibration of the cochlea vibrates the fluid, and when the fluid moves, since these hair cells are suspended in the fluid, the hair cells are going to move too. So when the hair cells move, they respond to that mechanical input, and the bottom of the hair cells right over here release neurotransmitters in response to those hair cells, those cilia moving. So that's when they synapse, yep, and then the electrical impulse is created and it gets sent down the auditory nerve.
Yeah, I just didn't get like where the outer hair cells, I missed that connection, but okay, thank you.
Yeah, no problem. That cleared it up for you?
Yeah, definitely.
Great. Okay, any other questions about that? Cool. So like I just said before, hair cells throughout the cochlea do differ in size, and therefore they register and therefore differ in the specific pitches they register. So I believe when you get to the very peak, right towards this inside, these hair cells are specific to lower frequencies. And as you start to move out towards this like outer curl, those are the ones that generate the higher frequencies. And those higher frequencies are the finer cells, the finer hair cells, and the finer cilia. So as you age, those are actually the first to go. It just kind of happens. The cells just through wear and tear, they'll start to damage and they'll start to wither away. So that's why as you get older, you start to lose the ability to hear these higher pitches. And so the ear is not only used for hearing, it also helps us balance. So I put an image down there. It's the same ear, but we'll talk about it a little bit differently. So the semicircular canals are important. They contain three. One of them is the utricle, one of them is the saccule, and one of them is the ampullae. So they're tubes that are filled with fluid, and they are located next to the cochlea. So if you look right here, these three tubes that are they just look like three rings that are sort of right next to the cochlea. Those are the semicircular canals. So those are super important in terms of your bodily awareness. So they contain hair cells as well. And the way in which they switch mechanical vibrations to electrical impulses is basically the same way. But instead of detecting sound, they detect the rotational acceleration of your head and linear acceleration of your body as well. So before I talked a little bit about the vestibulocochlear nerve and how it sort of branches off into two divisions once it gets close to the ear. So you can actually see that pretty well in this image. This yellow nerve is the vestibulocochlear nerve. And once it comes right over here, it actually splits off into two. So this part that's innervating the semicircular canals, we sometimes refer to as the vestibular nerve. And the one that's headed directly into the cochlear is called the cochlear nerve. Sometimes it's called the auditory nerve. Sometimes it's called the acoustic nerve. But these signals actually get transmitted to the brain through this same pathway once they converge. So we just call the entire thing the vestibulocochlear nerve. And the vestibular system communicates with the pons and cerebellum, and it helps maintain balance and equilibrium via that vestibulocochlear nerve. So we talked about the pons a little bit before. The pons and cerebellum are located towards the back of your brain. And just like the slide says, they are important for maintaining balance, equilibrium. They help you coordinate movements. And all of it is facilitated by the input of the vestibular system. Does anyone have any questions on that? Awesome. And if you think of any questions while I'm speaking, just pop them in the chat box. All right, so hearing loss. Whenever we get concepts that are generally conceptually easy, so like the pathways for vision and hearing, the MCAT likes to test like where defects happen in there. So I'll see hearing loss on it a lot. I'll see visual problems a lot, like nearsightedness and farsightedness. We'll come back to that in a couple of minutes. So I thought it would be pretty helpful to include this on the pathways for vision and hearing. So there are two major types of hearing loss. The first one that I want to discuss is conductive hearing loss, and it occurs when the mechanical parts of the ear are obstructed or prevented from transmitting sound to the cochlea. So that's pretty much anything in your outer or the middle part of your ear. So those ossicles, if anything happens with them, if there's any sort of blockage in your ear canal, if there's any deformity in the auricle, anything that prevents those hearing waves from reaching the tympanic membrane and being converted into electrical impulses would be conductive hearing loss. So, I mean, there are a couple of causes for it. They can be congenital, they can be caused by illness, by head trauma, Opal said, we have 14 cranial nerves. Do we need to know which nerve is the vestibulocochlear? You don't really need to know anatomy for the MCAT at all. You really don't need to be able to identify. Anytime the 14 cranial nerves comes up on the MCAT, it'll probably be in a passage. But you should know, I think it's a good idea to know that the vestibulocochlear nerve does innervate these systems, just like you should know that the optic nerve innervates your eyes. So you don't really have to know like the specific pathway, how they travel. You don't have to identify them, if they give you a map, like which is which, but you should definitely know that the structures that they innervate. No problem. Yep. Okay. So it affects the outer and middle ear, like I said, the auricle, ear canal, tympanic membrane, the ossicles, and it can be treated with surgery if there's corrective surgery that's available, or a conventional hearing aid. The conventional hearing aid will help amplify the sound and get it over to the tympanic membrane. And sensory neural hearing loss is the second type. So this occurs when the inner ear cannot successfully transmit electrical impulses down the cochlear nerve to the brain. So conductive hearing loss really focuses on not being able to transmit the mechanical information to the cochlea. And then sensory neural hearing loss, pretty much we're getting the information over to the cochlea, but there's a problem. There's a problem with the actual sensory neural. There's a problem with transferring the mechanical information into the electrical information and getting it to the brain. So this can be caused by aging, head trauma, disease, among a bunch of other different things. And obviously, since the nature of these two sorts of hearing losses are different, the treatments are a little bit different. So these treatments can be, they can include corticosteroids. Sometimes if it's head trauma or if there's a great deal of swelling, swelling can interfere with that vestibulocochlear nerve, and it can prevent information from getting to the brain. So sometimes they use corticosteroids to stop swelling. And there are cochlear implants as well. So I put the cochlear implant over here to the right, and it sits at the top of your ear. There's a transmitter that actually interacts with the cochlea. And this piece at the top, there's a microphone. The sound is input through the microphone. It goes through the processor, right up to the transmitter, and then that transmitter stimulates the cochlear nerve and the auditory nerve. So it doesn't allow the patient to hear completely perfectly, but it definitely allows for increased comprehension of speech. So, I mean, there are people that are not able to hear at all. They'll be able to, process certain sounds in their environment. And in combination with reading lips, language comprehension is greatly increased for those who are candidates for the cochlear implant. And I do see that pop up pretty often too on the MCAT. Does anybody have any questions about this slide? You can type in the chat or unmute. Cool. All right, so time to review again. So which structure is most directly responsible for converting mechanical vibrations into electrical impulses in your ear? So I'm going to set the poll up. You guys take a minute to think about it, answer it, and we will regroup in a minute and discuss. Okay, poll's open, so go for it.
Quick question.
Sure.
That's when you said corticosteroids. Do we have to know like the reason why they use them in those situations? And if so, what was it again?
So I would just say corticosteroids. Really, my knowledge regarding corticosteroids, whenever I hear that, I just immediately think they use them to treat inflammation. So anytime you're talking about a nerve, inflammation of the surrounding tissue can always impact the nerve's ability to transmit those electrical impulses. So sometimes when you experience like a super loud noise, those hair cells actually get extremely inflamed and the surrounding tissue gets inflamed. And if it's treated quickly enough with corticosteroids, you can actually save part of the nerve. So they do use that when it's, sort of like volume trauma or even certain types of head trauma. But all you should really have to know about corticosteroids usually is just that they treat it.
Just inflammation?
Yep.
Okay, cool. Thank you.
No problem. All right, take about 15 more seconds, and then I'm going to close up the poll and discuss. All right, get your answers in. I'm going to close it and share. All right, so we got quite a spread on this one. So 29% of you said A, 21% of you said B, 29% of you said C, 7% said D, and 14% said I have no idea. So the correct answer is actually C. And I'm kind of glad that it was kind of spread out, because I think this is a pretty good question, if I do say so myself, and I think it's a great opportunity to go over it. So again, which structure is most directly responsible for converting mechanical vibrations into electrical impulses? So most directly responsible, we just have to go through what each of them do. So the ossicles, it's the hammer, anvil, and the stirrup. Those transmit mechanical vibrations. So if you remember, the tympanic membrane vibrates and it vibrates onto the ossicles. Those ossicles vibrate onto the cochlea. So they're transmitting mechanical vibrations. The tympanic membrane, again, is just transferring sound in the air into the mechanical vibrations of the ossicles. The semicircular canals are basically like, they're similar to the cochlea in that they are fluid-filled canals that directly transmit vibrations in the fluid into electrical impulses. So just like the cochlea, when we get sound input, the vibrations travel into the cochlea, and then the vibrations of the fluid of the cochlea rub up against the hair cells. It's the same thing with the semicircular canal, only the semicircular canals actually, they track our movement. And the auricle, not many of you said that just because it's the outside of the ear. And I want to make sure you guys understand this. So does anybody have any questions about this? Because we can spend a decent amount of time on this. I think this is pretty important. The ossicles was definitely the distractor. I would just say that it's incorrect because it doesn't most directly convert it. It is involved, of course, and it's very close to the point where the vibration is converted into the electrical impulses. But if we switch over to the vestibular sense, the semicircular canals are actually directly implicated in transferring vibrations into electrical impulses.
Okay, this is Taja again. So the ossicles transmit, the tympanic membrane just like vibrates, or receives it. The semicircular canals, like, okay, so when I got to that part, I saw semicircular canals not directly like correlated to the cochlea. So would it be like on the same exact choices? Like would there ever be a question that says like A is the cochlea, like C is semicircular canals with this question?
I would not have done that because
I was like, it's the same thing, right?
Exactly. It's
Okay.
they're not the same thing, but they operate very similarly. So the
Okay.
semicircular, you could consider them like analogous structures. So they're
Okay,
operating
nice.
the same way, but they're transmitting different information to the brain. So the semicircular canals transmits like your balance information and the rotational acceleration and linear acceleration. The cochlea, in the same exact way, the fluid vibrating on the hair cells, it transmits auditory information. So that's why I knew it was going to be a little bit of a trick for you guys, just because whenever we're talking about mechanical vibrations, we're talking about auditory. But I just wanted you guys to keep in mind that the vestibular sense works in the same exact way.
Okay.
But yeah.
Thank you. Wait, so is that the reason why like when you go in a circle you get really dizzy? Is that that's this the
Yes, sometimes. A lot, and I think a lot of older people experience like fluid thickening in your ear. So sometimes, yeah, that can cause dizziness.
Okay.
So Alexandria said, how would that question be worded where A would be the correct answer? Could be worded in a different ways. I would say the ossicles are maybe one of the final steps of mechanical vibration. Maybe it would say like, what is the last structure, the last structure that information passes through before being converted, maybe. Just the giveaway for this is I said most directly converting into electrical impulses, because you should think about what information is coming in and how is the information leaving. So information is coming to the ossicles because the tympanic membrane is vibrating. So that's mechanical. The ossicles are then knocking on the cochlea and vibrating the fluid. So it's coming in as a mechanical vibration and it's leaving as a mechanical vibration. Whereas the semicircular canals, the actual movement of your head is the mechanical input, and then the fluid is rubbing up against the hair cells and converting it into electric. So it's coming in as that mechanical movement and it's leaving the semicircular canals as an electric impulse. So yeah, it would be pretty helpful to solve this question if you look at it as how is the information coming in and what form is it leaving in. And the only one here where it comes in and it leaves as a different type of information, a different type of medium, is the semicircular canals. Yeah, no problem. All right, Opal, what's your question? Do you have a question, Opal?
I thought I'd unmute. So I had gotten it hit in my ears maybe 5 years ago. I was playing around and then I had to see an ENT doctor. So I have a deviated septum and it affected my breathing. And it's, so you had mentioned a structure that connected the ear to the throat. Why would my breathing be compromised from being knocked in the ear?
Well, because I mean, my guess would just be that ear, nose, and throat, obviously they're all connected. I mean, the nose and throat more obviously are connected. And then, like we said before, the structure you're referring to is the eustachian canal. So its primary function is just to connect the ear and the throat to equalize pressure between the two. But I mean, they're all pretty connected. They're all in close vicinity. So any sort of trauma, any sort of hit to your head, it's going to affect them. I mean, they're pretty delicate structures. You see these tiny bones in your ear. You could do some damage with a pretty big hit. In terms of breathing, I think the deviated septum probably had a really big role in your breathing. Was that your question?
Yeah, I just wanted to know which structure. You said it was a eustachian canal that connects.
It's called the eustachian tube, yeah.
And it connects the ear to the throat. Okay.
Yeah, so like whenever you feel your ears pop, that's because that's the eustachian tube connecting and equalizing the pressure.
Okay.
I heard a tilted atlas could cause tinnitus in one of your ears. I'm not sure. That would stink if that's true. Okay, does anybody else have any questions about this before we move on? Okay, great. Okay, so now let's get on to the mechanism of vision. So I put this little graphic over here. It is your eyes and your brain from the top view. I just wanted to put this on here because I didn't mention elsewhere in the slides the optic chiasma. It's also sometimes referred to as the optic chiasm. And the main takeaway of this picture is just that there's a crossing over right here before the visual information reaches your brain. So this is color coded very nicely so you can logically follow. If you look right over here towards the top, your left visual field is in red. So you can see that it's actually the right side of the both eyes that are responsible for your left visual field because your vision is actually flipped. And you could see those red when you follow it down. When you follow it down these nerves right here, you can see that there's a crossing over of the red and blue right here. So anything in the left visual field is going to be processed on the right side of your brain, and anything on the right visual field is going to be processed on the left side of your brain. So this crossing over is what enables us to see one continuous image. That's why both of your eyes, you can see the left visual field, and both of your eyes, you can see the right visual field, as opposed to your left eye only seeing the right visual field and your right eye only seeing the left visual field. So that's something I don't see it come up too often, but it's really cool, and I think you definitely should know it because it could pop up on the MCAT, and it's not too advanced of a concept. And then you could take a look at more of the structures over here in your free time. Here's the lateral geniculate nucleus that we were talking about, which is located in the thalamus. You can see that it stops over there, and then it gets sent right over to the primary visual cortex. So the structure of the eye, again, there's a lot of information on this slide. I don't think it's too conceptually hard, but is a lot of terminology that you should definitely be familiar with. Excuse me. So light enters the eye through the cornea. Ruth said, does this mimic the concept of which light passes through converging lenses or concave lenses? Very interesting. We're going to talk about that in a little bit. So optics actually does come into play when we talk about like deformities of the eye. So we're going to talk about nearsightedness and farsightedness in a couple of minutes. And that actually converging lenses and concave lenses do come into play. So hold off for a couple of minutes and we're going to touch on that in a little bit. But for right now, let's just look at the structure of the eye. So light enters the eye through the cornea. That's that first layer right over here. It's usually curved. Sometimes if it's curved too much or too little, we have problems seeing. The cornea is continuous with the whites of the eyes, known as the sclera, and that's just, you know, the whites of your eyes. It's continuous throughout, and it's continuous with the cornea. So light passes, after it enters the cornea, it then passes through the pupil, and it reaches the lens. So right over here. So it passes through the pupil, and this white thing right behind the pupil is known as the lens. Excuse me. So the lens further refracts it, and it fine-tunes it before it reaches the retina. So there's a lot of refraction that happens with the light that enters your eye before the retina is ready to receive the information. And the curvature of the lens is varied by the ciliary muscle. So I believe it's on, yeah, it is. So the ciliary body and muscle. So that's kind of like the iris. I put the iris down here as well. They're muscles. The iris is around the pupil and the ciliary muscle is around the lens. So they can contract and they can relax to allow different levels of light pass through and focus different levels of light on the retina for you to get the clearest image. And obviously problems with vision occur when you know, the cornea is too curved and the iris can't accommodate for the pupil, or the lens is too curved, you know, and the ciliary muscle can't accommodate and can't focus properly onto the retina. So after it passes through the lens, the ciliary muscle, light passes through the vitreous chamber. The vitreous chamber is just fluid. It's just fluid that is like a medium for the light to pass through to get to the retina. And then it reaches the retina, and it's basically just the back of the eye, and it's covered in a bunch of photoreceptors. So we have two types of photoreceptors as humans, and they're known as rods and cones. We're going to talk a little more in depth about them on the next slide. But that's basically the path that light takes through your eye. Yeah, it goes through the cornea, goes through the pupil, passes through the lens, goes through the vitreous chamber, and then the image is focused on the retina, and the rods and cones will receive some of the information, and we'll talk in a little bit about what happens after that. So I just put these down here just because I didn't mention them when light, you know, like the series of light passing through, but I still think they're pretty important to know. So I already said the iris is a muscle that controls the amount of light coming through the pupil, and the macula is the center part of the retina. It has a high concentration of cone cells and is responsible for our central vision and the high visual acuity in the center of our visual field. So when you stare directly at something, you're going to be able to see a lot more detail at the thing you're staring directly at, as opposed to something that's in your peripheral vision. And that's because the macula is the center part of the retina. Even more central in the macula is a part called the fovea centralis. And that is where super visual acuity occurs, when you're staring super closely at something, and you can see all the fine details. And that's on the next slide as well. Opal, did you have a question?
Not at all. Did I?
Okay, I just saw your hand raised. Okay, never mind.
Oh, let me lower it.
Okay. Does anybody have any questions about this before I move to the next slide? Okay. All right, so a little bit more on the photoreceptors that we were just talking about. The photoreceptors in our eyes are known as rods and cones, and they are found distributed throughout the retina. So like we said, the retina is the back part of your eye, and there are rods and cones that are all over it. So your rods work in low light environments. So they detect basic properties about what you're seeing. They detect size, shape, contrast. They bring in black and white light, basically. So they do not detect color. They do not detect fine detail. That's why in low light environments, when you're out at night, you can kind of see, you can't really see color very well, but you can see shapes, you can see contrast. We have a lot of them, especially compared to cones. There's 120 to 130 million in humans. Yeah, so that's a lot. And cones are the other sort of receptor. These are the more fine-tuned photoreceptors that help us see fine detail, help us see color. And they require adequate lighting to function. So there are about 6 to 7 million of them in humans. And like I said before a little bit, most of them are focused on the macula and specifically in the fovea. So the fovea centralis, also sometimes I've just seen it written as fovea before. It contains a large concentration of cones and is responsible for the extreme visual acuity that you utilize when you're staring directly at something. So you have the retina, which is the entirety of the back of your eye. Then you have the macula, which is central to the back of your eye. And we have more visual acuity there. And then in the middle of the macula, you have the fovea, which is super visual acuity, a bunch of cones there. We can see a lot of detail. We can see a lot of color. But of course, we need light to see that. And then the only other structure that I would say is worth mentioning towards the retina is called the blind spot. It's also known as the optic disc. So this is where neurons converge to form the optic nerve, and there are no photoreceptors present there. So there actually is, all day, every day, we're using our eyes, and we don't really notice that there's a blind spot, but you actually always have a blind spot in your field of vision. Your brain is just very, very good at convincing you that it's not there. I've seen on a couple of study materials, they'll do like little demonstrations where they'll put 2 dots on the page. They'll tell you to cover one eye, look at the dot, and then as you bring your head farther away from the page, the other dot disappears, which I think is a really good example of how the blind spot is always there and we never really realize it. So if you have some free time and you're interested, you can definitely look it up. I'm sure you'll be able to find one all over the internet. Does anybody have any questions about photoreceptors, about the blind spot, the fovea? You can unmute, you can use the chat box. Okay. All right, so let's talk a little bit about the defects of the eye. So these 3 terms, definitely know. They're the scientific terms, and then I just put the normal person terms right next to them. So emmetropia is also known as normal vision. Light is properly refracted onto the retina. So this is sort of where the lens shape comes into play, like Ruth mentioned earlier. So if you take a look at this normal eye up at top, you can see what's happening. These 3 are just representative of, the photons that are coming in, the light waves that are coming in. So this light should converge at one specific point on the retina. And the problem with nearsightedness and farsightedness is that this image is not converging directly on the retina. So myopia is nearsightedness, and that means that the light is refracted too much, resulting in a focal length that is too short. So as you guys know, I think we did a light in optics. It might have been last night. It was definitely a past lecture. Anytime light passes through a different medium, it has that index of refraction. It's going to change. It's going to bend depending on the medium that it's going through and depending on the shape of the lens. So if you look at this nearsighted, this diagram right over here, you see that it's converging too quickly because the focal length is too short. So this image is not being properly projected onto the retina. So, I mean, it's because the cornea is too curved. So when the cornea is too curved, it actually shortens that focal length. So, I mean, the ways to treat that is you can either flatten the cornea or you can shorten the distance of the eye. You're just trying to bring this focal point to reach the retina because that's the problem. Yeah, so I think I just said that actually after I saw your question, Opal. That's how myopia is corrected. Usually, I think in practice, the way that it's actually corrected is that the cornea is flattened. I think that's the easiest way to increase the focal length so that it actually reaches the retina and it produces a clear image. And then on the other side of the spectrum, we have hyperopia, which is farsightedness. So light is refracted too little, and it results in a focal length that is too short. So if we look over to the right, this light is not being bent enough. So by the time it reaches the retina, it's not converging into a singular image. So you're not going to be able to see properly. And it's the exact opposite of myopia, nearsightedness. Farsightedness, it's either because the cornea is too flat, or it's because the actual length of the eye is too short. So if the length of the eye is increased, or if the cornea is curved more, the focal length is going to decrease, and it's going to bring this actual focal point onto the retina, and it's going to help focus the image. Okay. Does anyone have any questions?
Yes, Chris, I have a question. God, why don't I do this? So I'm thinking.
I think you're breaking up a little, Opal.
I know, I'm trying to just not get the echo.
Sure.
All right. If I wanted to correct it, the spreading, as in you said nearsightedness, it's refracted too much. Why is it doing this?
I think someone else needs to mute their
I'll get back to you.
Yeah.
Yeah, can you guys all make sure you're muted? Okay, you were saying? All right, you still there, Opal?
Yeah, but my phone, I'll ask at the end once I figure out how to silence this echo.
Okay. Okay, so maybe we can talk about that later or during office hours if you're having issues right now, or we can, yeah, okay. So give me 1 second. Sorry guys, hold on. Okay.
I think I figured it out now. So Chris,
okay.
Can you hear me now?
Yeah, yes, I can.
Yes?
Yes, yep.
Okay, so my question is, if in myopia, you said the light is being refracted too much, so it looks like it's hitting the retina, if it's converging before it hits the retina, based on your photo, right?
Yes, correct.
It stops short of the retina before it hits the retina.
Yep, because that curvature, it's too
doesn't
much
he give you like a lens that will spread it out, like a diverging lens, so it doesn't hit the retina prematurely?
Yeah, I believe that's how they do the lenses. When you look at the MCAT, at least in my experience, they're probably going to ask more so, like when it comes to defects of the eye, they'll probably ask how you can actually alter the eye, like the structure of the eye specifically, rather than using glasses. At least that's what I've seen the most. So, I mean, as long as you have a sound understanding of how optics work, which it seems like you do because you were just explaining it to me, you'll be good. So you can see the nearsighted eye. The cochlea is, I'm sorry, not the cochlea. The cornea is too curved. So it's a converging lens, but it's converging too soon, like you said. So we have to kind of find a way to, diverging lens to try to expand that and increase that focal distance so that it actually projects onto the retina. So yes.
Or in your case, you said you just flatten the, what do you, flattening?
That's how they do it. They flatten the cornea. I'm pretty sure that like LASIK eye surgery, when they're correcting nearsightedness, they flatten the cornea. And it helps. Yeah, and it's pretty very minimally invasive, and there are very good results with it.
Got it. Thank you.
Yeah, no problem. And yeah, I would encourage you guys to even do some research on LASIK eye surgery, maybe even just a few minutes of looking it up, because I have seen it come up a couple of times on the MCAT. they'll always explain it to you in context, but I think it's a good idea to have some background information on that as well. Same thing with the cochlear implant. I've seen cochlear implants pop up on my studies as well. Okay. How does astigmatism affect the eye? So, jumping ahead a little bit, but astigmatism is basically when the cornea, it doesn't really have much to do with the curvature being too much or too little. It's sort of a deformity in the cornea. So it's not really the focal length that's the problem. The problem is that the actual cornea is not shaped correctly, so the image just has a hard time focusing at all. It's not really the focal length. And that's pretty much what you have to know about astigmatism in the scope of the MCAT. Okay. So we're going to do another review question. So what change would need to occur in an emmetropic eye to make it hyperopic? We just talked about this a little bit, so see if you can remember, see if you can reason. Take a minute or two. I'm going to open the polls now, and we will discuss in a few minutes. Okay, polls are open. All right, take about another 15 seconds, get your answers in. Okay. All right. So 54% said A, 38% said B, 8% said C, and nobody said D or I have no idea, which I like. So the correct answer is in fact A. Great job. It's funny because you will usually see questions that give you a hyperopic eye and say, what do you have to do to make it emmetropic? So I switched it and I wanted to see if I would fool you. And apparently I did not fool the majority, which is great. So like we said before, the emmetropic eye is able to focus. It has the right focal length. So hyperopia, you just have to remember that that's farsighted. If you have a hard time remembering it, just remember that hyper means a lot, large. It's a large distance, it's far. So hyperopia is farsightedness. And in order to get farsightedness, we have to increase that focal length. So the point where the image converges is behind the retina. So in order to do that, when we take the curvature out of this lens, it takes a longer distance for this image to converge, and it ends up doing that behind the retina, and that results in hyperopia. So if the cornea was made curvier, that would result in myopia. The image would be focused before it reaches the retina. If the length of the eye from the cornea to the retina was made longer, again, that would result in myopia because we're having the image, it's focused before it actually reaches the retina. And D, the cornea should be distorted to an irregular shape. that wouldn't cause hyperopia, that would cause astigmatism. It would just be a blurry image that's not able to be concentrated. Does anybody have any questions on that? So we understand that the major factor that comes into play here is the focal length. So the two ways that you can adjust the focal length in regards to the retina is that you can either flatten or make curvier the cornea, or you can increase or decrease the length from the cornea to the retina. We understand that?
Yeah, Chris, there's on your slide, the previous slide, the definitions are the same for hyperopia. Look at your slide. Could you go back to the previous one?
Sure.
You have the near side is light that's refracted too much, resulting focal length that is too short. And it's the same definition.
Gotcha.
Hyperopia.
Okay. Yeah, so I'll fix that. Okay.
Short.
Good catch, Opal. I'm glad you're paying attention. So I will fix that before we put it up.
So that's why I chose the other answer.
Gotcha.
Focal length is too, I guess it wouldn't matter.
Okay, that's understandable though. So I'm definitely going to fix that. Thank you for pointing that out. So the hyperopia at the bottom,
what's the proper definition of hyperopia then? Is the focal length too short or is it too long?
It's too long. So if you look right over here to the right, the farsightedness, we can see that it converges after the retina. So that length, the focal length is from the cornea from when it starts to converge to when it actually converges. So we can see that that focal length is too long. It's by the when the light gets to the retina, it's not converged yet, so it's going to be blurry. So I will. So
the answer will still be using a flat.
Correct. Yeah, because when you
still using a. What's the answer for A again? What's the answer?
The cornea should be flattened.
Okay.
Yeah, because that increases the focal length, so that would make the eye hyperopic. But thank you for pointing that out. I'm going to correct that. Okay. Yes. So right down here, the focal length, it should say that is too long. Myopia, a myopic focal length is one that is too short. Alexandria said, what occurs if the cornea is curved? Your cornea should be curved. Naturally, it should be curved. The problem is if it's curved too much or if it's curved too little. So when it's curved too much, we're nearsighted. And when it's not curved enough, we are farsighted, because too much of a curve results in this short focal length, and too little of a curve results in this too long focal length. So if that's a little bit fuzzy, I would encourage you to go back to the optics lesson. Study optics a little bit more. It will greatly help your understanding of this stuff. And Jessica said, how do they correct hyperopia? Do they curve the eye more? I mean cornea. Yep. Yeah, you can curve the cornea. You can, or decrease the length of the eye. Anything, I mean, and it's pretty intuitive when you look at it. So you're trying to get this point that's not to the retina yet, you're trying to get it to the retina. So what can you do? You can either move the back of the retina up to that focal point, or you can change this cornea over here so that it takes a little bit farther of a distance to focus, and then it actually reaches the retina and it looks like the normal eye. So yes. All right, so we're almost done with the lecture. I just wanted to throw this in here just because it's kind of relevant for sensation and perception when we're talking about pathways in vision. And I do see this pop up decently often, sometimes on multiple sections of the MCAT. So we're going to talk about the two different types of thresholds that usually come up. I know some of you have probably seen this even in psychology when you do perception for cognitive psych. So the absolute threshold is the minimum intensity needed by a stimulus to activate a sensory receptor 50% of the time. So that means it is how you can detect that a stimulus is present, the minimum amount of stimulus intensity that is needed for you to be able to recognize that the stimulus is present 50% of the time. That's considered the absolute threshold. So anything under it, you're pretty much not going to be able to recognize it. Anything over it, you'll be able to recognize it, because 50% is that sort of that chance number. Anything over it is not by chance. Anything under it, again, is not by chance. And the difference threshold is just comparing two different stimuli or a change in the stimuli. Is that related to the absolute refractory period for the action potential? I wouldn't say necessarily. I mean, you might be able to draw some parallels between the two. The absolute refractory period just basically has to do with the inactivation of the sodium channels. And then just the complete hyperpolarization of the membrane, where this absolute threshold is, I mean, I guess you can kind of relate them because it does have to do with action potentials. They're sort of paralleled. Yeah, I would say they're sort of parallel because it does depend. You need 50%. Activating a sensory receptor 50% of the time would require the action potential to fire. So it's a good idea. You can draw those parallels if that helps you remember them. And the difference threshold is another threshold, but it's not just we're not just detecting a stimulus. We're detecting a change in a present stimulus, or we are distinguishing between two different stimuli. And it's also known as the just noticeable difference. I think I've actually seen that terminology more often on the MCAT. And that's actually usually the threshold that I see, because you have this nifty little law called Weber's law that gets a little bit quantitative, and they like to test it because it's one of those simple laws where they'll throw numbers at you and you should be able to do it without a calculator. So Weber's law is delta I over I equals K, which is pretty simple. Delta I is the actual difference threshold. So that is the difference between the initial stimulus and the change. It's the actual change is the difference threshold. And I'll explain it in a minute. I is the initial intensity of the stimulus, and K is the constant value. So if you have, say, a weight that's 1 pound, and you take another weight that's you add 0.2 pounds to it. Maybe you don't feel the difference when you add 1.2 pounds to it. You add 1.3 pounds, you finally feel it. So the delta I, the difference threshold, is going to be 0.3, because that is the difference at which we can finally distinguish the difference between the two stimuli. Yeah, so, and then I is the initial intensity of the stimulus, and K is a constant value. So if you have the same thing, if you have those weights and you increase them, say instead of starting with 1 pound, you start with 30 pounds. That means that the delta I is going to change. But if you take that delta I and you divide it by the original I, that K value is going to be the same as if the I was 1, if the I was 10, if the I was 100.
So a quick question. Sorry,
Yeah.
this is Taja. So different show, like I'm a very visual person. So I'm kind of and like experienced kind of teacher. So
Yeah.
so kind of like warm, the difference between warm and like that will burn my skin off, kind of like that.
Yeah.
Okay.
Temperature is definitely something that can be measured with Weber's law. Yeah. That's actually very interesting because I don't see that too often. I usually see it with weight or sound. So Opal said, will the units be log scale like decibels? Yeah, I mean, they will use decibels. Sometimes I think I've even seen them use just like absolute units for sound, like even just like made up units. But I often see Weber's law used with pounds or some sort of weight, just because I think it's just easier,
Yeah.
because they know you don't have a calculator, so they want to give you something that you're capable of doing in your head. So for all recorded values of I and delta I, K will remain constant. So I did want to do a review question on that. I know nobody wants to do math right now, but I want you guys to get this, and then we'll go over it. So so we can do it in practice and really understand what Weber's law is, because I think it's definitely important for you guys to know. So read this, take a couple of minutes, and then we're going to go over it. Let me open the polls for you. Okay, polls are open. All right, I'll give you guys about another minute. And I'll give you like a 10-second reminder. All right, guys, get your answers in. Okay, I'm going to close the polls. Nice. Okay, so 0% said A, 7% said B, 87% said C, 0% said D, and 7% said I have no idea. So that is great because the correct answer actually is C. And let's definitely go over this. So I put the work at the bottom. Give me 1 second. Okay, so the difference threshold for detecting additional weight to a 5-pound weight is 0.5 pounds. So what that means is that if I'm holding a 5-pound weight, they're telling you that I will not notice a difference. I will not notice that this weight has gotten heavier until you put 0.5 pounds on it. Anything under 5.5 pounds, I'm not going to notice a difference. Once it hits 5.5, then I can finally distinguish that there's a difference between the two of them. So what Weber's law is saying is that if you divide that 0.5 by 5, you're going to get a constant K. So in this case, when we divide 0.5 by 5, we get 0.1 or 1/10. So that K constant is going to be the same no matter what intensity the stimulus is. So we can change that I to any number, no matter what that is in relation to K, what that is in relation to delta I, the K is going to stay the same. So now that we figured out that our K is 0.1, we find the second value for delta I. So I equals 12. So we know I equals 12. We know K equals 0.1. We just plug it into Weber's law again. So delta I over 12 equals 0.1. We move the 12 over to the other side. Delta I equals 1.2 pounds. So in practice, that means that I'm not holding a 5-pound weight anymore. Now I'm holding a 12-pound weight. I'm not going to notice that there's a difference in the weight until you add 1.2 pounds. So until that 12-pound weight becomes 13.2 pounds, my brain won't be able to register that there's a change in stimulus. Does everyone understand that? We can definitely go over this because I really would like you guys to know this. Okay. Feel free to use the chat. Feel free to unmute. An everyday example of Weber's law.
What I mean is,
yeah.
When will I not know that I'm holding something that's 1.2 pounds heavier? Why would I not know if I'm holding something in my right hand and then something in my left that's heavier? Why would the brain? I'm just trying to figure out how is this realistic?
Right. So it's basically, so don't think about it. Yeah, kind of. So don't think about it as holding two separate weights in one hand. Think about it as, say someone is trying to like use you for an experiment and they're trying to figure out how much, like how you can notice how the weight in your one hand is changing. So say you stick your hand out and someone gives you a textbook that's 3 pounds. They're going to add a weight that's super, super tiny and they'll ask you if you recognize it. You're not going to recognize it until it reaches that difference threshold. So say the difference threshold for that is maybe a half a pound, we'll say. So if they're adding a 0.25 pound to that 3, if you're not looking at it, they're going to say, have I changed anything? You're not going to recognize it. If they put 0.4, have you changed anything? You're not going to recognize it. So once they put that 0.5 or whatever weight it is, once you finally recognize that there was a change to that one hand, you finally recognize that there was a change in the stimulus, that's the difference threshold.
Mm-hmm.
Mm-hmm.
Okay, so it's not separate hands. It's like me holding a textbook and you putting like a
yeah,
feather on top of the textbook.
exactly. They're trying to see, it's how you can perceive the change in that specific stimulus.
Okay, and question, when it comes to dampening sound waves, like, do we adapt after a while when you hear loud music, you kind of just get attuned to it? What's going on? Is that the threshold effect?
I think that's more like desensitization.
Okay.
Like for perception, that doesn't have much to do with the difference threshold, but it does occur. That is something that happens.
Thank you.
Yeah. Okay. Yeah, so this is the last slide of the lecture. So if you guys have any other questions, feel free to ask them. You can type them in the chat. You can unmute. Other than that, we're finished with the content part of today. So I'm going to make some adjustments to the PowerPoint. I'll send them in the GroupMe. We'll upload them in the OneDrive folder. And they should be on YouTube. The full video should be on YouTube tomorrow, and we're also uploading them in segments as well. And that should be up within the coming weeks as well. So if you guys don't have any questions, that concludes the lecture today. I'll stick around for a couple of more minutes. Other than that, happy Friday and happy studying.